Hello, Dr. Murugan. Can you hear me?
Yes, Professor. Good afternoon. Students, we'll be entering our third lecture for the day. Today we have with us Professor Deepshika Chakravarti. She is a professor at the Department of Microbiology and Cell Biology in the Institute of Science, Bangalore, India, and currently holds an Astra Chair professorship. She is an Humboldt Fellow and did her research work at FAU Erlangen, Germany. Professor Deepshika did a master's in microbiology from Nagpur University and a PhD from National Center for Cell Science, Pune. Her research focuses on infectious disease and vaccine development. Her work has been published in top biological journals, including Journal of Experimental Medicine, EMBO Journal, Journal of Immunology, Infection and Immunity, the Journal of Clinical Microbiology, AAC, Journal of Antimicrobial Chemotherapy, BIIS, Pathogen, etc. She has won many awards to her credit, like Tata Innovation Fellowship, Astra Chair Professorship, SK Chatterjee Award, DAE Outstanding Investigator Award, National Bioscience Award, DBT, NASI, NASI, Reliance Industries, Platinum Jubilee Award, etc. She is a Fellow of National Academy of Science, Indian National Science Academy, and Indian Academy of Sciences. She is involved in the editorial board of many journals like Virulence, Innate Immunity, BIIS 1. She serves as a reviewer in a reputed journal like Lancet, Nature Communications, MMBR, to name a few. Her research has been supported by grants from the Department of Science and Technology, Department of Biotechnology, Indian Council of Medical Research, DAE. She has more than 150 papers in the international journals and has guided 20 students so far towards their PhD, and 10 more are continuing towards their PhD. More than 200 summer trainers and 60 academic summer trainees have worked in her lab. Most of our PhD students are faculties in reputed institutes like IIC, BITS Pilani, NIT, etc. Professor Deepshika is a passionate teacher and mentor, and she aims to teach microbiology to the children and general audience and to bring out the importance of microbes in day-to-day activities. So, this is a short introduction. I invite Professor Deepshika to deliver the lecture.
Thank you, Dr. Murugan, for quite an extensive introduction. Thank you so much. It's my pleasure, as usual, to talk to you young colleagues. And today I'm going to tell you a story of one of the very important arm of the innate immune sensing system. I think you can ask me questions in between also. Please unmute yourself and ask. And if you do not want to, please put your questions in the chat box. And in the end, we shall be taking all the questions. Yeah. But please ask, because the interaction is extremely important. If you're not able to, understand anything, kindly ask. I hope you can see my screen.
Yes, Professor.
Okay. Can you see the full screen mode?
Yes.
Okay, great. So, yeah, again, welcome colleagues and good afternoon. I have taken, I think, 1 or 2 lectures in this platform, and it was really very good. Lot of encouragement, because the students did ask very important questions. And I came to know through this one lecture that I took that the basics of immunology is kind of very unclear in, in many of the sectors. So I was not very sure if the same batch of students are attending also this particular class, but I thought that, batch. Is it the same batch, Doctor?
Different batch.
A different batch, okay. So now what we will do is I'm going to take you through one of the very important, let me just put my pointer. I'm going to take you through the very important component of the immune system, which is innate, a bridge between the innate and the adaptive immune system. So when you look into the components of the immune system is so robust and the immune system is so beautiful that it can distinguish and act both in a non-specific manner and in a specific manner. We will come to this one by one. Get into the basics, colleagues, so that if you have any further questions, it will be very easy for you to understand and get it clarified. Now, when I talk about the non-specific and the specific immune system, both of them have two different arms. One is a humoral arm and one is a cellular arm. Here also, specific immune system will have a humoral arm and a cellular arm. Now, what do you mean by the word humor? How does this word humoral come from? Humoral means it's a fluid, anything which is secreted. That's a humorous, in the humor. Now, in this humoral arm, you have a complement, you have interferon, tumor necrosis factor, many other cytokines. In the cellular arm, you have macrophages and neutrophils. Now, as you know, that the macrophages are one of the most important cell type that takes up everything. It just goes around and it takes up everything that it sees next to it. It just phagocytoses, a very efficient phagocytic cells, and a kind of a gatekeeper and a scavenger who will go and scavenge everything to make us safe. Similar cases with neutrophils. The neutrophils are one of the most potent cells to just degranulate itself and release its toxic content into the environment. Very important. Specificity does not come into picture here. Anything that it senses that it is not right, it simply opens it up and degranulates itself, which gives rise to the increased content of, say, granzymes and other antimicrobial peptide into the surrounding. When it comes to specific immune response, you have again a humoral and cellular system. And in this case, for humoral, it is the antibodies. You know what antibodies are. They are made by the cells which are called as a plasma cells. Normally, when I ask students who makes antibodies, they will blatantly tell B cells. Remember, it is not the B cells. The B cells mature to plasma cells, and it is the plasma cells which makes antibodies. Yeah. Then the cellular part are the T cells. Of course, the B cells and the other cells do come into picture, but the T cells and the other effector cells, they are the part of the cellular immune response, which is very specific. Now, how the body in fact gets protected? It is a very fine balance, a very intricate balance between the infection and the immunity. Now, if your balance is toppling this side or that side, will determine the course of your action. So a very perfect balance between the infection giving rise to a correct kind of immunity is the one which protects us. But as you know, the pathogens are too smart and too clever, because if body was supposed to take care of the pathogens, none of us would get sick, because the cells are many, systems are many. You know that that does not happen because the pathogens are becoming too clever now. The evolution of the pathogenesis in every single pathogen, be it bacterial or be it viral, is so robust that I am very surprised that where we will see the end. Forget about the antimicrobials and the new antibiotics in place. There is actually no end. So we need to, kind of retract ourselves and understand what do we really understand by this infection and what is this immunity. Now, if anybody gets infected, all of us get infected by various bacteria, various virus. All of you have survived the COVID phase, and many of us, I do not know how many of you have got infected with the COVID. If the innate immunity is very strong, we will come to, that is what we are going to discuss. How important is the innate immunity for you to immediately attack and give it a kind of a standing time for the body to respond to get into the adaptive immunity phase. Now, if the innate immunity is very robust, in fact, the person will not get the disease. It will be cleared off at the stage of entry itself. But suppose if the innate immunity is not very strong, then what happens? Then the disease will be there, but this disease may not be fatal because the adaptive immune system will kick in. Now, the innate immune system is very robust. It will also crosstalk between the adaptive immune arm. This is known as an innate adaptive crosstalk. And the disease cannot be established. But suppose if the innate immune system has not kicked in very nicely, the person will suffer from the disease, But then the recovery can set in because the adaptive immune system has kicked off. Now, when the adaptive immunity is full blown, and even then the reinfection happens, there will be no disease because there will be some memory cells which will kickstart the process of providing the immunity so that the person will not suffer from any disease. But if the adaptive immune system fails and there is no memory, then there is no rescue. And that is what is the death phase. So the response to infection, if you look into this particular cartoon, is dependent on two major arms. One is the innate immunity, one is the adaptive immunity. And the interplay between these two will decide the course of action. Now you will ask me, then what is the reason that we suffer from bacterial diseases all the time? What is the reason that we suffer from influenza virus all the time? Because the reinfection actually gives rise to the similar disease. That means that in few diseases, there is no memory response. This is a very big arm in the immunology where all the vaccinologists are trying to understand how do you boost up the memory response so that this reinfection does not happen. It's a very tricky question. Not all the bacterial disease will give rise to memory. And that is the only reason one suffers from diarrhea, dysentery all the time. You can get infected with salmonella and suffer from typhoid fever or gastroenteritis all the time. Same is with Shigella, same is with mycobacteria. Why are bacteria so worse in triggering the, the innate, the memory response? The smartness lies in the bacteria. That is the reason where they are very successful pathogen. Influenza virus, they keep on changing their H and N positions, and that is how you do not get any kind of protective response and no memory there. So in all these cases, what do you do? Because every time the infection sets in, you need to do the entire course of innate, adaptive, clear the infection, get rid of it. Again, it comes, again, you have to do the entire. It's a big waste for the body to actually do that. Now, the significance of the immune systems are indeed many. It is only because of them that we are safe. Now, mind you, colleagues, I will just take 1 minute here and take a pause. Your immune system, how good it is, depends on a very good gut flora. I will just bring this small word of gut flora because always remember to take care of your flora. The immune gut axis is very robust. So do not, under any condition, abuse your gut flora. The moment that gets abused, your entire immune system collapses. Okay, so kindly take care of your gut flora. Now, the beneficial effect of the immune system is definitely it will protect you from the invaders, and it will also be very beneficial for the elimination of the altered cells. That means if some of the self molecules become a little different and behave funny, the immune system can take care of that. But the same immune system becomes detrimental. You all have noticed it specifically during the COVID time, that the person was not dying because of the virus itself, but because of the immune system going haywire. And that is the detrimental system where your immune system goes haywire, out of control. And this word have become cliche, which is known as a cytokine storm. Beyond the capacity for the body to take care of these large amount of cytokine, they give rise to this storm. So this leads to the discomfort, that is the inflammation and damage to the cells. So the lot of autoimmunity sets in. Along with these two, now you know that the cytokine storm, which is not at all good for the body, actually can take a life of a person, leading to septic shock. So the death is not due to the virus or the bacteria. The death is because too much of cytokine in the body by these immune cells, leading to the septic shock. So everything has a balance. It's a fine tuning of your immune system. Too much of immune system, very detrimental effect. Too less of the immune system is again a detrimental effect because infection takes the upper hand. So we have to keep a balance. To know more about the innate and the adaptive immunity, you need to have a very good understanding about the characteristic. You know, immunology is one field which rapidly progresses. And because of this very progressive nature, there are many things which are continuously changing. And that is the reason why immunology is such a demanding subject, and you have to really keep up to date with the literature. Now, innate immunity definitely is antigen independent. What does it mean? That means you do not need a very specific antigen to kickstart your innate immune response. As the name suggests, it is innate. Innate means everything is inbuilt. Anything comes in, it has to respond. That means that gatekeepers or the armed forces must be ready to attack whatever comes in, irrespective of whether it is a bacteria or a virus or a parasite or a fungi. It has to attack. So it is not dependent on the antigen. Whereas the adaptive immune system is very much dependent on the antigen, because this adaptive system is now kickstarting a very specific response. And that is the reason it has to be antigen dependent. There is no time lag. The body or the system cannot afford any kind of a time lag to respond when the foreign harmful person comes inside the body, and the response time. So if the time lag is there, by the time the pathogen is so smart that now also it is doing, in spite of having a very robust system and in spite of having no time lag, pathogens are smart enough, and that is the only reason we fall ill. Now the evasion strategies are so smart. Only when you understand this characteristic, can I tell you more about the ways by which it tweaks these evasion strategies. So there is no time lag for the innate immunity, but you have a big time lag for the adaptive, because if you do not have an innate, adaptive system cannot kickstart. So the bridge between the flow of information from the innate to the adaptive makes it a bit late to come into the picture. This is not antigen specific. As it is not antigen dependent, so it is not antigen specific, whereas your adaptive immune system is antigen specific. And there is no immunological memory. Memory means that there are some cells in the immune system, once it encounters the antigen, will keep the memory of that antigen pattern. And that memory is very important to give rise to a full-blown immunity when the second reinfection comes in. In innate immunity, there is no immunological memory, but in the adaptive immunity, there is a development of memory, and this memory development is a hallmark of a good vaccine. If a vaccine is not able to give memory, then you can imagine how many shots we have to take to get rid of or to kickstart the infection, the immune process. But having said that, the no immunological memory in case of this adaptive innate immune response is changing now. There are a couple of reports which mentions that the dendritic cells, the DCs or the dendritic cells, which are the major antigen presenting cells, they do have a memory. Slowly this field is emerging. As I mentioned to you, immunology is an ever-changing field. We need to be really up to date with the literature because of the fast-moving speed of this particular area of science. So there is a bit of a hint that the innate immunity do have a kind of a memory. Adaptive immunity memory development is a very long-studied one, and now we are trying to understand that why these bacteria do not lead to the development of or suppress the immunological memory. This is the evasion strategy of a pathogen. Now, the components of the immune systems for both adaptive and innate are extremely important, because you need to go layer by layer, right? It takes a lot of layers for a pathogen. So the life of the so-called pathogen is also not light. It's extremely, stringent. It's very difficult. But how do they do this? In spite of all that, we become sick. That means we need to really understand the smartness of the pathogen. Each pathogen, it depends on what pathogen you are targeting. That is why I cannot be generalized in saying. If suppose the crowd says that many of you are working with mycobacteria, they have a far different strategies than salmonella or say brucella or say legionella. Okay, so the pathogen that you are studying, you need to really get into the depth to understand that after all these layers, how they still make us sick. Now, the physical barriers in case of innate immunity can be many. All the coverings from skin to the gut villi to the lung, the ciliary actions of the lung, the nasopharyngeal cavity, everything is a physical barrier. which is going to take out the pathogen and try to restrict it at the physical barrier. But when you talk about adaptive immunity, there are none. There are no such physical barriers when it comes to adaptive. So all the informations are fed from the innate system to the adaptive system. The soluble factors that are there in the innate immunity are many proteins and many non-protein secretions, which can bind to the pathogen of interest like complement. It can bind it and it can get opsonized, then it can be taken by the macrophages, degraded, etc. The TNF secretion, it can kickstart another pathway, signaling pathway, and take care of the pathogen. So there are many soluble factors. In case of adaptive immunity, you have immunoglobulins, that is the antibodies, and of course, many of the cytokines. Now come the cells. In innate immunity, you have phagocytes. As I mentioned to you, these are the ones which goes and moves around. You know, like a person who is going around and making sure that everybody is safe. So the phagocytes are extremely important cell to go around. Macrophages are one of them. So you have tissue-specific macrophages. So for example, in liver, the macrophages are known as the Kupffer cells. So the Kupffer cells will go and make sure that the liver is safe. In the blood, you have something called monocyte. Now, monocytes are monocyte macrophage kind of lineage, where the monocytes will go and get into the tissue and become macrophages. Macrophages are the final cells which will take up. Then the NK cells, the eosinophils, the K cells, each one of them. In case of adaptive immunity, the two very important cell types are the T cells and the B cells. And I am sure that a bit of your knowledge in the BSc, you should be knowing the ontogeny of the lineages of the T cell and the B cell. You must also read, it's very important and interesting to read that how the T cell diversity and the B cell diversity came, and must not forget to read Susumu Tonegawa's Nobel Prize winning work to understand the B cell and the T cell diversity. And that is the reason we are actually so safe, because everything can be taken care of due to this diversity. Now, the effector mechanisms in the innate immunity depends on the site and the components that it has. So for example, the skin, the squamous cells, the first cell, if anything happens, there is a desquamation and there will be flushing. There are sweat glands, there are many fatty acids which secretes, which all take care of the pathogen. In the gastrointestinal tract, you have the columnar epithelial cells. There is a peristaltic movement. Because of the peristaltic movement, very low pH secretion of the bile salts and the fatty acids, the pathogens cannot actually establish. So the pathogens needs to make a very strong effort to withstand this peristaltic movement, which they do by some other ways. But mostly from day-to-day life, the body can, the GI tract can take care of any, bacteria or viruses or parasites which comes inside. Then in the lung, everything that you inhale gets into the trachea, the ciliary, the cilia and the trachea with its sweeping movement can take care and get rid of it. So mucociliary elevator, surfactants in the lung, all of them, they take care of the any foreign particles that come in. Now, nasopharynx and the eye, which are a very protective site, and you know that it is very seldom that you suffer from a serious eye infection. Some of us do. If our eye become very dry and we are not shedding tears or saliva in our oral cavity, during those times, the infection rate is high, because the lysozyme in the tears and the flushing action, it takes care of all the bugs that are there. You know that lysozyme is very important enzyme, right? And where do you use lysozyme? You use this lysozyme in making your plasmid preparations many a time, because it acts on the peptidoglycan layer. It cleaves your beta-1,4 glycosidic linkage between N-acetylmuramic acid and N-acetylglucosamine. And it is there in your tears. Now, the blood and the other lymphoid organs, you have a phagocytes, NK cells, natural killer cells. The phagocytic activity and the intracellular killing is extremely important. You can have a direct or an antibody-dependent cytolysis of the cell which is harboring the pathogen of interest. Then you have a serum and other fluid in which you can have lactoferrin, transferrin, which will quench the iron. Now, iron is a very important element for pathogen. Pathogen requires iron. So many of the pathogens, they will quench it from the host. So what lactoferrin and transferrin will do, it will not make it available because it has quenched the iron. So it make it iron deprived. So the pathogen will be iron deprived. And if it is iron deprived, it is not going to proliferate and establish well. Then you have interferons and the TNF alpha, which will have the antiviral proteins. It will activate the phagocyte, etc., etc. I mentioned to you lysozyme. It will hydrolyze your peptidoglycan. Then the fibronectin complement will lead to the opsonization, enhanced phagocytosis, and then it will make the macrophages to take up the foreign particle, degrade it with the help of the lysosomes inside the macrophages, thereby clear the pathogens. Okay, so now the innate and the adaptive immune response are very vibrant. So we must understand what kind of encoding of the receptor is. In case of innate, it's a germline encoded, whereas in case of adaptive, it is somatically rearrangement. So this is why I told you that kindly go through Susumo Tonegawa's Nobel Prize winning discovery, which talked about a lot of these the rearrangements, which can give rise to both the T cell diversity, receptor diversity, as well as the B cell antibody diversity. Now, the distribution of the receptors, this is not clonal, this is non-clonal, whereas in adaptive, it is clonal. From one, you will get many, so it is clonal. Now, the repertoire of receptors, how many receptors one can have? In case of innate, it is very limited. But in case of adaptive, it is large. Due to this diversity, it is very large. And that is the reason it can take off almost every antigen, in fact, which comes on their way. Now, the target, in case of innate, is invariant. But in case of adaptive, it is very much variable. The recognition is extremely perfect in case of innate. It can be a bit of imperfect. And that is the reason why in adaptive immune response, you can have something which is known as a self-immune response, which leads to autoimmunity. That happens during developmental stage when certain cells go haywire and they very sneakingly escape the teaching phenomena, you know, the self versus non-self. So it can be imperfect, and that leads to a long-term a problem which is known as autoimmunity. In this speed, when you talk about the speed, the innate immunity is extremely fast because there is no lag phase. The adaptive is slow because it has to develop. Long-lasting memory, as far as the research goes, it is no at the moment, but there are a couple of research which is saying that some subset of the DC, they do tend to show some memory response. In case of adaptive, definitely yes, they do have a adaptive, they have a memory response, but then it depends on the pathogen. How do they help them to develop the memory response? Because the memory is the most important part of the immunity. It is the most important part of a successful vaccine. Otherwise, we will keep on taking vaccine after a couple of months, and there is no memory to protect us from the future challenges. What is the determining factor about your entire immune system and which makes you safe is the crosstalk between this innate and adaptive immune response. Now, you know that your immune system is a very energy exhaustive process. You understand that? And that is the only reason you must not tinker with your immune system, must keep it very safe through keeping your gut flora safe. And it should not, you know, just simply go haywire and start kicking your immune response when it is not required. So the induction should only happen when there is an appropriate antigen, which belongs to a pathogen. Now, suppose if an antigen which was enclosed, which is a part of the cell, somehow got exposed to the body, and then it starts making your antibody, that is where the problem happens, which leads to autoimmunity. So induction is very important. It should be very appropriate. Now, activation of the lymphocyte for any self-antigen is extremely deleterious. And now you also know that the major cause of the morbidity and the mortality in this modern world are the two metabolic disorders, which is there in almost every household. One is diabetes and another is hypertension. Both of them are stealthy. Both of them are kind of absolutely obnoxious when it comes to the problem that it causes. And now diabetes is known to be due to these self-antigens against the beta cells. So the role of autoimmunity in diabetes is almost proven. So the beta cells, the pancreatic beta cells, which are the major cells which produces insulin, the antibodies develop against that. And that is how the beta cells are destroyed. So you can imagine that how deleterious it is if it starts making the antibody. Similarly, muscular dystrophy, making it against your own muscle protein. So the adaptive immune system must have the signal, that is knowing that from where your antigen is coming and what kind of response will you be inducing it to. Now, the signal which tells the adaptive immune response that, okay, you need to now make the system ready, will be provided by your innate immune system. And that is why the bridge or the crosstalk is extremely important. Now, the first interface is between the innate and the external environment. That first interface from the environment is fed into the innate immunity. Always remember, colleagues, this basic you must remember. There is no jumping from the environment to the adaptive immunity. It always has to cross the innate immunity. And that is why keep your innate immune system very strong so that you have a subsequent stronger adaptive immune response. Now, the first interface by the innate immune response group is done by the pattern recognition receptors. The pattern recognition receptors, surface receptors, and the effector molecules, these play a hallmark role in determining how robust the phenotype will be. Now, the innate immunity or the innate response feeds into the adaptive immune response. This is the second interface. And then there will be many other receptors, which we will study in detail. But the pattern recognition receptors are something which had made a multi-billion dollars. I'm sure you might have heard of it. It is known as a pathogen-associated molecular patterns, which are recognized by the pattern recognition receptors. Now, how it is, you just pay your attention to this cartoon, okay? Now, the innate immune cell is this on my right-hand side. APC means antigen-presenting cells. One of the very important antigen-presenting cells is none other than the dendritic cells. Okay, colleagues? So remember this. APC is nothing but dendritic cells. Now, the PRR is pattern recognition receptors, which are present on the dendritic cells. Now, this APC is going to make your this pathogen recognized by the PRR. Then it will transduce the signal and it is going to present this antigen. It will make this peptide antigen. It will load it onto the MHC class and it will be making loading this to the MHC2. Now, this particular peptide loading will be recognized by one of the cells. In this case, it is the Th2 cell, which kickstart the Th2 arm. This is the second signal. Now, this will be recognized by the T cell receptor. And this the T cell receptor, when it recognizes the antigen which is loaded onto the MHC, they will make a bridge. Now, this bridge alone is not enough. Okay, this bridge gets strengthened by this co-stimulatory molecule like CD28, and this bridge which recognizes the peptide antigen onto the MHC2, TCR recognizes it. This bridge gets strengthened by this particular signal. So these signals are called as a co-stimulatory signal. When this entire, you call this as an immunological synapse. You might have heard about neurological synapse. This is called as the immunological synapse. Now, this synapse is extremely important for the cells to kick-start their signaling process. Now, when this synapse is stabilized, the Th2 cells start making interleukin-4, interleukin-13. These are the anti-inflammatory signal. And this then take care of the adaptive response. Now, if it is a Th1 cell, then they will make a lot of IL-2, then IL-6 and other cytokines, TNF-alpha, and then they will take care of the adaptive immune response. So these two molecules, CD28 on the Th2 cells and B7 of the APC, these are called as a co-stimulatory molecule. So signal 1 and signal 2, both these signals are required to make a very strong immunological synapse to give rise to the cytokine response. I hope that this is very clear now. Okay. So now, a minute. Okay. So the mechanism of the immune recognition, which is done by the help of this pattern recognition receptors, now they should be ready-made, okay, onto the surface of the cell. The pathogen-associated molecular patterns, they are the one which are recognized by the PRRs or the pathogen recognition receptors. Now, how very few receptors can recognize many pathogens? This was the first question that late Professor Dr. Charles Janvey Jr. asked. He called it as the immunologist's dirty little secret, that when every cell type can be removed, still how the body can mount and respond. And he realized there is something known as the pathogen recognition receptors, which recognizes the PAM. Now it is very unique, and it is the evolution of the innate immunity is driven towards recognizing this invariant molecular constituent. So a very limited number of these germline encoded receptors, they are able to recognize a wide variety of these molecular structures. We will see what these structures are, because they make a combination. So for example, if you have a toll-like receptor 1 to toll-like receptor 11, they will make a combination between 1, 2, 2, 4, 2, 6, like that. So various permutations and combinations, you can do your math and figure out how many different signals it can recognize. So the pattern will be recognized by this pathogen recognition. You can have many kind of pathogen recognition receptors like toll-like receptors, nod-like receptors, lipopolysaccharide binding protein. And the culmination of this reaction is to induce the inflammation cytokines to destroy the microbes. So the pattern recognition receptors can be divided into two kinds. One is a endocytic PRR, second is signaling PRRs. Now the endocytic PRRs, they are very classical, very nice PRRs like mannose receptors, the scavenger receptors, whereas the signaling pattern recognition receptors are the toll-like receptors, which made a big havoc in the field of immunology, and the industries minted a multi-billion dollars, by making an agonist and antagonist for these TLRs and the NLRs. Now, what are these TLRs? The toll-like receptors, as I have mentioned to you, they are present on the membrane, on the plasma membrane. Some toll like TLR1 or the 6 or the 2, 4, 5. There are certain toll-like receptors which are not present onto the membrane to begin with. They are present on the endosome, like TLR3, TLR7, TLR9. Apart from these TLRs, you have something called as a NOD, nucleotide oligomerization domain. That is what NOD stands for. Now, the NODs are known as a cytosolic sensors, whereas these tolls can be there on the surface of any cell type, and they can just sense all the pathogens that are coming. For the NOD sensors, they are only present in the cytosol. So if they escape the TLR and they somehow come to the cytosol, NOD will come into picture. So the NOD has these kind of domain, card domain, NOD domain, the leucine-rich repeat domain. Now this leucine-rich repeat is also present in the TLRs. This is a hallmark of the identifying that how many TLRs do you have. What are these PAM, pathogen pattern? You can have lipopolysaccharide from the gram-negative bacteria, peptidoglycan, which is found, lipotecoic acid, mannose-rich glycan, flagellin, pillin, anything and everything, double-stranded RNA, almost all of them, they can be recognized. Now look here, this is a lipopolysaccharide, which is present only in the gram-negative bacteria. They can bind to the LBP, lipopolysaccharide binding protein, that can be recognized by the TLR4. Mycoplasma's lipopeptide can be bound to the TLR2 or the TLR2, TLR6 heterodimer. Bacterial lipopeptide can be bound to the TLR2, TLR1 heterodimer. Similarly, double-stranded RNA can be sensed by TLR3. There are flagellin can be sensed by TLR5, bacterial flagellin. Similarly, single-stranded RNA and some chemical compounds can be sensed by TLR7. Bacterial CpG DNA can be sensed by TLR9. So all these TLRs are ready-made. You don't need any lag time for them to make. Anything which comes gets sensed by the TLRs. Now, what happens when they sense? So this is called as a toll-like receptor signaling pathway. Here, the major science goes in. It's a very interesting science, which can tell you to tweak any kind of this toll-like receptor with the help of its antagonist to make your inflammatory cytokine more or less. If you tweak any kind of this toll-like receptor, the downstream signaling goes through the adapter molecule called as a MyD88, finally going through a lot of these transcription factors through the NF-kappa B, very famous, well-celebrated transcription factors, and then it can go to making of the interferons, cytokines, etc., etc. But where is your first signal coming? The first signal is coming from the pattern recognition receptors. So these were the receptors which I told you, they are present on the surface. Now, if the bacteria gets hijacked inside and they escape all the other pattern, the cytosolic sensors which belong to the node sensor, they will come into picture. Get a broad idea about all these sensors so that you can fine-tune your thinking later whenever you're working with a pathogen. So the node sensors, they can be categorized into many kinds. Now there is an endless list of the new node sensors which are coming in. So for example, node 1, they can recognize the gram-negative bacteria, mesodiaminopimelic acid. The node 2 can recognize both the gram-positive and the gram-negative, both kind of bacteria. The muramyl dipeptide, which is a part of your peptidoglycan, can be recognized from both the gram-positive and the gram-negative. Now you have another group which belongs to the lab, like IPAF1, NALP3, the DAI RIG, all of them, they are now known to recognize many, many different PAMs which are there. Look at the list. Don't even bother to look at this small written list because the list is never ending. But the research is very interesting because every day that a new kind of a sensor gets recognized, it is a kind of a hope for us that the body can still fight a lot of other pathogens. Now, these are the sensors like CARD domain. CARD stands for caspase activated recruitment domain. Yeah, so node 1 has a 1 CARD domain, node 2 has a 2 CARD domain, etc. And they have many different adapters. So these cytosolic sensors are indeed an amazing sensor which can take care of the pathogens. So for example, there was a beautiful paper in Nature by Takawaka et al. Way back in 2007, which showed that a double-stranded DNA can be sensed by this DAI. It's a cytosolic sensor. They still don't know what the adapter is, but the DNA can bind, the double-stranded DNA can bind to this cytosolic sensor, thereby leading to the activation of the NF-kappa B, and so is the CpG DNA. They can trigger through the TLR9, they can also lead to the NF-kappa B, and thereby leading to the transcription of a lot of these interferons. Now, the similar way by which the node 1, how do you remember what is the difference between node 1 and node 2? Actually, it's very simple. Node 2 has a 2 CARD domain and it can recognize both gram-positive and gram-negative, whereas node 1 has a 1 CARD domain and it only recognizes the gram-negative bacteria. Okay, so if you now look into this signaling pathway, it is so complex, right? The bacteria coming in, the peptidoglycan, the flagellin, cell's own molecule like uric acid, the ATP, bacterial RNA, viral RNA, each one of them, when it is there in the cytosol, it triggers a group of a sensor. And this sensing molecule will give rise to many of the cytokines. Now, here is a problem. Somebody who is suffering from a lot of uric acid issues, you know the gout disease, uric acid, you have a pain in the leg also, or many toxin accumulation, or high amount of ATP, if it gets leaked from the cell is going to sense it and will give rise to a haywire immune system, which is very detrimental for you. So that is the only reason why we should be very sensitive to our immune system and try to keep that in the check. I think my time is, okay, I have few minutes now, then I will leave it for question and answer session. So the node will sense the bacterial infection. Say, for example, node 1 will sense the Shigella, E. coli, Pseudomonas, Helicobacter pylori. And the node 2 can sense both Streptococcus pneumoniae as well as the Mycobacterium tuberculosis. This is also very nice paper. If you get a chance to read this paper by Akira in Nature, where they have mentioned about the role of the RIG1. This is a retinoic acid inducible protein 1 and a melanoma differentiation associated gene, MDA1, as an intracellular pattern recognition receptor. So in 2006, RIG1 and MDA1 got added up to the list of these so-called node sensors. And they are able to distinguish between the different RNA viruses, and they are going to appropriately induce the antiviral response. Now, that may be the only reason that why viruses, we can get rid of them quite fast. And the viral immunity is long lasting because they have a very good memory response, except few viruses like influenza virus. For that matter, even COVID, they may have some kind of immune, memory response, but we still are taking now third booster, fourth booster. We don't know because the virus is very new. So if you see this one, on the one hand, the TLR9, the TLR3 can also recognize the viral RNA, and the viruses which can send out their genetic material can get recognized by a lot of these other, not cytosolic sensors. Each one of them will crosstalk, but ultimately will either go through NF-kappa B or will go through this IRF signaling pathway to give rise to the cytokine response. Now, this is a small glimpse I gave for the virus. The same kind of glimpse is there also for the bacteria. Same is for the fungi, and kind of similar response for the parasite. Now, in spite of having all these responses, the pathogen's ability to evade the system is something that is very interesting. I will open it up for question now. I have ended my talk. So let us open this up for discussions. We have 15 minutes. I have kept almost 15 minutes for discussion. And we will make sure that the discussion continues very well. Yeah. So colleagues, kindly start the discussions.
Students, you can ask questions.
Yeah. And I hope that we have something in the chat box. Let me check it out. If there are questions in the chat box, I will keep this open so that there is nothing in the chat box.
So far.
Okay, colleagues, please open up, unmute yourself, put your video on, please introduce and ask.
Ma'am?
Yes, hello.
Hello, I'm Kadambari from Anna University.
Yes, Kadambari. Please go.
You mentioned that the immune system, that the gut flora was a major, had a major role in the immune system.
Yes.
The gut flora is being explored a lot now with several systems. So
Yeah.
what aspects are interacting with the gut flora to mediate immune system?
Now, because now the gut flora is a much talked word, and more we know, actually, it is like the less we are understanding it, because the papers are many. The gut flora, which is the consortium is very wide. It is more than 230,000 different species. The product of this gut flora are small molecules, fatty acids and very small molecule, which can cross the barrier and interact with many cells. So that is how you have a gut-brain axis, you have a gut-liver axis, you have a gut axis, which is itself there. It is called as a gut-associated lymphoid tissue or mucosal-associated lymphoid tissue. Now, the flora matures or modulates the system by giving these kind of small molecule signals to the immune cells. And that is how the immune system gets matured. Now, as good the system of maturation in the immune cells happen, the better it is for the system not to fire unnecessarily. And that is where the most important point comes, that how good one is in not allowing the immune system to be firing it unnecessarily. where the person then suffers from inflammatory bowel disease, right from inflammatory bowel disease to the cytokine storm. Yeah, so that is how it is. Just not the gut flora, but also the mucus, that 100 micron thick mucus, which is there beneath the gut flora. So both of them should be preserved very carefully for the good immune system, for the maturation of the good immune system. Okay, Kadambari?
Yes, thank you.
Anyone else?
Yes.
Hello, ma'am. I
Hello.
had a doubt.
Yes.
Ma'am, in your section, you told about the vaccine's ability to produce efficient memory.
Yeah.
So, like, is this why for like cold vaccine we are taking a dose 1, dose 2, and booster vaccine, or is it like against different variants?
Yeah, good question. Now, why are we taking this booster doses? Variants are coming in. The doses has nothing to do with the variant, to tell you the truth. Now, if we have taken Covishield dose 1, because the virus is new and we do not know how we are responding in terms of its memory, we are taking a booster. Many people have measured their antibody titer and the antibody titer seems to be excellent. Now you are talking about the variant coming in, and the booster doses are not being done with the variant. They are done with the same vaccine. So is it really going to help us? The answer is no and yes, both. If the variant is being made in the spike protein, which recognize the antibody, then we are gone. Then it is of no use. But if the variant is coming, which is different from the antibody binding site, where the neutralizing antibody can bind and neutralize the virus, then it is great, because whatever variant comes in, the antibody binding site is intact and the antibody can come and neutralize it, bind it and neutralize it. But it is too early to tell what is going on. And at this time, now the government has given a mix and match. So one can take 2 Covishield followed by a Covaxin or a Moderna or a Pfizer. I think now government is allowing it, and hopefully variant vaccine will come. But first of all, we should know that what is the status of these variants? Do they have the same mutation where the antibody binding site is mutated? Then it is a very bad story. Then we need to come up with the new vaccines. So at the moment, yes, it is just to make sure that your memory is intact. And any kind of reinfection with the COVID will lead to neutralization of the virus due to the availability of the antibody in the system. Okay, Sarita?
Yes, ma'am. Thank you so much.
Yes, welcome. Yes, colleagues, candidates, students, please ask. Don't feel shy. Just ask, because it's very important to ask. And remember, nothing is trivial. Even knowing what is the toll-like receptor is not trivial. So shed it. We are anyway online. So, and you are behind the screen. There's no need for you to feel shy. Just keep asking. Good afternoon, Shaukat. Yeah, please.
Ma'am, actually, like in coronavirus, we are looking at all the different variants, like they are improving. But was it the same earlier, ma'am? Like in the past, there were many diseases and all. So variants were also improving like this, or this is something very new?
No, Shaukat, this is not new. It is just that our understanding has improved so much. Now, if you talk about the Spanish flu, that also have taken millions of lives and somehow it had faded. Now, you don't hear any deaths due to by the same strain, right? Now came COVID. Now, this COVID virus, which actually belongs to the Coronaviridae family, will definitely come with the variants. There is nothing new because viruses are known to evolve. But it depends on the type. Now, if you talk about smallpox virus, which was deadly once upon a time, chickenpox, which was deadly too, all have the vaccine and the variants have not come in. They are not very prone to change. So one infection with chickenpox gives us a lifelong, long-term memory, right? So you do not hear any deaths. Smallpox is eradicated. No more smallpox. What about these viruses which are coming up? Same is with Ebola or the West Nile. They come, they kill and they go. They are kind of endemic. When it becomes pandemic, flu sometimes is a pandemic, corona is a pandemic everywhere. It's going into different hosts, getting challenged by different environment, making a variant. That is the nature of the virus. Now we are only knowing now because it is hit hard. So we are knowing now variants are coming. Sequencing facilities are very good. You can give it for sequencing and you know that variants are coming, which is the nature of the virus. But the question is, where is this variance going? Is it making the virus more fitter, or is it making the virus more attenuated? As per the data goes, and if you really look into the cases of hospitalization versus death, it is trying to give us an indication that it is making it really, really milder, which is very nice. So the variants are becoming milder, and most probably the antibody which is generated due to vaccination scheme is good enough to neutralize them. And thankfully, these variants are not being made at the same site where your antibody will bind. Then we are gone, yeah? But they are very milder, so we don't have to. But as far as your answer goes, they were there. It's just that we are trying to understand this, as the tools are wonderful now. You can just study them. Sequencing facilities came a long way. And robust sequencing facilities, the PAN program, that's all working very good here.
Yeah. Yes, ma'am. So, ma'am, can we expect that at some time they will stop evolving the variant?
Never. There are many sites, there are many residues which can be prone to getting evolved. But at the same time, if that tug-of-war ends and if the virus somehow tries to get that understanding, then in that case, they may stop. But I can never think, if that would have been the case, no, influenza virus would have stopped evolving, but it is not. Every season comes, you have a newer version of influenza virus. Yeah.
Thank you, ma'am.
Thank you. Yes.
Next question.
Just anything in the chat box. Dr. Murugan, if anything is there in the chat box, kindly alert me.
Okay.
Yeah. Till now, there is nothing in the chat box. We do have 15 participants. Few of the colleagues have asked. Rest of them are very quiet. No more burning questions?
Hello.
Yeah, hello.
Good afternoon, ma'am.
Good afternoon.
Ma'am, I'm Apurva from NIT Warangal.
Yeah.
So, ma'am, I have a question regarding methodology.
Okay.
For example, how do you approach a research problem? In the sense, for example, if you have a fungus which is attacking a particular plant, for me as an undergraduate, I'm not exposed to many knowledge, and I don't even know what are the methods I can use to prevent it. So how do you suggest a person like me approach a research problem? Because for this BIIS, we are supposed to come up with a research proposal. So I was wondering, how do you do that?
I don't know. I should ask Dr. Murugan to give me another slot to just for this, for all of your benefit, to say that why and how and when you should do your research. And indeed, you asked a very important question. How do you approach a research problem? Because the research problem is something which is not trivial. If you can answer 3 questions, first question, which is the most difficult to answer and you really, really need very deep understanding, is a why. Why you would like to do this study? Not because your mentor is saying so, or your senior has left a piece of the work or something, or you dug it out from literature. No. Why is it that you would like to do this? What is putting the intriguing in that question in you that is making you to take up this project? It can be a very basic question, but if you are able to understand the why, then answering how and when becomes very simple. But the question why is the most important. The first key for approaching the problem is knowing the subject. You cannot, under any condition, dig out any problem or formulate any question if you do not know ABCD of that particular subject. The depth of readership and up-to-date literature is the most important point here. And that comes with the help of your mentor. That also comes with your own interest, of your own understanding. You cannot simply pick up words from the literature and say, okay, this is my project. No. The deep understanding, which takes actually years of readership. Now you are an undergrad student, right? You need to rely on your mentor to figure out that why, which have interested you, is, are you in a right direction? You may not be, right? How will you know? Because you have not spent that much amount of time in research or guiding student than your mentor has. So here the mentor plays an extremely important role, extremely important. And without the mentor's guidance, please do not even try to move forward. You should come up with ideas, but always vet it.
Right.
Not it, Apurva?
Yes, ma'am.
All the naive questions which I have faced during my undergraduate class in ISC or elsewhere across the world have actually led to one of the most fruitful research projects, because these are very important. But the student who is asking the question must understand that it is so important that I will take it up and move forward with the help of, again, with the guidance of mentor. Once you spend some 1.5 to 2 years, then you are kind of, you know, your path is set. You will exactly know how to go about it. But you definitely need a mentor alongside continuously to guide you through.
Right, ma'am.
Right,
Thank
Apurva?
Yes, ma'am. Thank you.
Any other questions?
Looks like there are no more questions. Is it true? No more questions about anything, be it your science or your career or the subject? Doesn't look like Dr. Murugan.
Ma'am,
Yes.
one more question.
Yes, Apurva.
So this is again regarding to my career. So as you know, because of the pandemic, I didn't get too exposed to any lab techniques or anything, you know, very practical. So I would definitely like to study further, but I'm not very sure of which area I'm interested in. You know, I am doing 2-3 projects, but then since everything is remote, it's not going very well. So what do you suggest I do?
So why it should be remote? I could have shown my lab. I'm in the lab only. I have 15 trainees. Half of them are from ICERs. 1 or 2 of them are doing their MS project. So many of them are here. Why you are doing remote, I don't understand.
Ma'am, for example, this.
Remote at all now. This is all in person. How you can do it remote, Apurva? You are in NIT Warangal.
Yes, ma'am.
Why is it remote here?
Ma'am, for example, actually, this particular project I was relying, this particular internship, BIIS internship, I was, looking forward to, but this is now online. And the one project I'm doing is in bioinformatics, so that doesn't really need to be in lab.
No, that you need not worry about so much.
The workshop is only online. If you want a training, if you want any kind of trainings, you are anytime welcome to our lab. That is already mentioned.
Yeah, I think you should get in touch with Dr. Murugan. Maybe you people do not have the real idea of what is going on, because and you are saying no, that I don't know, I don't have my experience. All the students who are here in the lab, half of them they don't have, because anyway they have to come and learn. So it's okay to be very fresh and come and learn, rather than learning something wrong and then coming and de-learning things. So they don't know, they just come, they nicely learn, and then they get trained in a project. First question you ask, I don't know what I want, that I cannot answer for you. That you have to be first clear about what you want. How will you get cleared about what you want? Is just ask yourself what you enjoy reading the most. Forget about practical training. I will tell you my own story. Till my PhD, I did not even know what Eppendorf is. I was so, like jumping with joy first time for my PhD. I hold Eppendorf in my hand, pipette, and saw Eppendorf tubes, so cute, tiny tubes. So nothing matters. We never had any exposure. The question here is not about exposure. You are not a technician, right? To do technical work, your thinking have to be developed. Now, what do you enjoy reading the most? What do you relish the most? That you ask yourself. Then you apply. You will be fine. Yeah, you need not know anything. As I mentioned to you, we are all growing scientists. So you will learn wherever you go. There is no need of any pre-learning, pre-hand exposure because somebody will teach you.
Right, ma'am. That's nice. Thank you.
Just change the thinking. I think it's a wrong thinking. The same student today morning, he came and told me, Madam, due to this remote instrumenter, I don't have. I said, good, you don't have. At least you have with blank paper. It will get absorbed like blotting paper now, which is good for us. Yeah.
Any more questions? That's
yeah, looks like there are no more questions, Dr. Murugan.
yes.
So if there are no more questions, thank you, colleagues. Thank you very much for this opportunity. Thanks, Dr. Murugan, for this nice opportunity.
Thank you so much, Professor, for your time.
And hopefully, why there are only 14 students and all others are joined live stream or something?
They have taken permissions to
Okay.
take the offline questions.
No, maybe they can be enthused to ask.
If we were there, some are taking permissions.
Sure. Thank you very much. Bye-bye. Take care, all of you.